Okay, this presentation is going to cover the cell cycle, and it is by me, Ashlyn Partain. The importance of cell division. Cells have to divide, and that's it. There are many reasons for this. One of them is to replace old damaged cells, and another one, this one may be more important, is because cells cannot grow indefinitely. This because if they grow indefinitely, then the proper nutrients won't be able to exit the cell or enter the cell in the proper amount of time. What I just explained to you is represented by surface area to volume ratio. The surface area to volume ratio decreases as the cell gets bigger. Interphase. Interphase is a major part of the cell cycle. It's actually where the cell spends most of its time, although it is far more simple than mitosis, which I will cover most of this presentation over. There are 3 stages of interphase, G1, S, and G2. G1 is relatively simple. The cell just performs normal activities and grows. In the S phase, synthesis occurs and DNA is replicated. One way you can remember S phase is because synthesis starts with a S, and it's the S phase. Finally, the cell is prepared for mitosis in the G2 phase of interphase. Mitosis in animal cells. Although mitosis is shorter than interphase, it is far more complex. There are 4 stages of mitosis in animal cells. They are prophase, metaphase, anaphase, and telophase. One way you can remember these stages is by remembering PMAT, P-M-A-T. In these stages, 2 identical nuclei are created before cell division. I would also like to note that there are slight differences between plant and animal cells, and I will be showing you the animal cell. Time in each stage. The following pie chart depicts the time each phase of the cell cycle spends in each stage. As you can see, interphase is much longer than mitosis. Interphase actually covers 3/4 of the cell cycle, while mitosis only covers 1/4. Okay, stage 1 of mitosis, prophase. 4 different things occur in prophase. Before I start explaining what they are, I would like to explain my picture. So, as you can see, the dotted circle is the nuclear envelope, and the reason it is dotted is because it is breaking down. And the chromosomes are represented by the X's, and there's a reason for their size, which I will explain later. And the spindle fibers are connected to the centrioles, and they are the little thread-like things. Okay, so in prophase, the nuclear envelope is broken down, which is why it's dotted, and that prepares the cell for nuclear division. Centrioles are also moved to different sides of the cell. The chromosomes condense, which is why they are so big and bold, and the mitotic spindle is formed. Stage 2, metaphase. In metaphase, the chromosomes are aligned in the middle and are thus attached to the mitotic spindle fibers. One way you can remember metaphase is because meta rhymes with middle, and the chromosomes are aligned in the middle. They are moved to the middle in a cellular form of tug-of-war. The duplicated genetic material carried in the nucleus of a parent cell is separated into 2 identical daughter cells. In anaphase, the 3rd stage of mitosis, the cell's spindle fibers shorten, thus forcing the sister chromatoids to move to opposite sides of the cell. So the spindle fibers are trying to stay attached to the outside of the cell, and to stay attached while they're shortening, they have to pull. And since the chromosomes were attached to the spindle fibers, they're pulled with them. And what used to be represented as X, the chromosomes that used to be represented as X, are now half X's. This is the phase that actually separates the duplicated genetic material into 2 identical daughter cells. Telophase, the 4th stage of mitosis, is the beginning of the reformation of many parts of the cell. The nuclear envelope reforms, which what we saw in prophase, it was breaking down, so now it's reforming. but of course there are 2 cells now. The chromosomes, which used to be represented as X's and half X's, are now dispersed into chromatin, the squiggly line in the middle of the nucleus, and the spindle fibers dissolve. It is the beginning of visible cell division. Cytokinesis is when the daughter cells physically divide. It is the end of cell division. The word cytokinesis literally translates to cell movement. Once mitosis and cytokinesis are complete, there are 2 identical daughter cells. One way you can remember the 4 stages of mitosis and cytokinesis is by this cheer. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis.